If you've ever opened a research paper and thought, I want to understand this, but this is dense, then this podcast is for you. Every day, thousands of research papers are published. Most of them are rigorous, insightful, and almost impossible for non-specialists to read comfortably. But the ideas inside them shape medicine, technology, business, and society. Welcome to One Paper Orbit. We are a research paper podcast. Here, we take peer-reviewed research and turn it into clear, structured conversations without adding speculation, without exaggeration, simply making the original work easier to understand. Today, we are discussing a paper authored by Qingqing Zhao, Chen Gu, Guoshui Luo, Shuwan Dai, Dan Wang, Xifei Zhang, Feng Wang, Hui Xu, Yanbing Han, and Xitao Wang, titled Combined Multi-Omics and Brain Pathology Reveal Novel Biomarkers for Alzheimer's Disease, published on 31 October 2025 in Springer Nature. This discussion is based on a paper published under the Creative Commons Attribution 4.0 license. The original authors retain credit for their work. This podcast provides an adapted explanation for educational purposes, with full citation and license information provided in the description.
Yeah, and the scope of what these researchers are trying to tackle in this study is just incredibly ambitious. I mean, they're trying to map the unseen mechanisms of a disease that affects millions.
Right, and they're doing it by processing just an almost unfathomable amount of biological data.
Exactly. It's a massive undertaking.
And that mission is really vital for anyone listening today. I mean, Alzheimer's disease accounts for at least, what, 2/3 of all dementia cases in people over 65?
Easily. It's the vast majority.
Right. And while we know its physical hallmarks, those famous amyloid-β plaques and the neurofibrillary tangles that build up in the brain.
The stuff we've been studying for decades.
Yeah, exactly. The exact underlying mechanisms driving the disease are still, largely mystery to us. So the goal for today's deep dive is to unpack this study that uses a really high-tech multi-omics approach to hunt for completely novel biomarkers.
Yeah, and to truly appreciate why this paper is so groundbreaking, we have to look at the specific tools the researchers brought to the table here.
Proteomics stuff.
Right. Historically, a lot of biological research has relied on looking at just one piece of the puzzle at a time, single omics. But here, they are layering different types of massive biological data sets directly on top of each other.
Okay, let's unpack this concept of single omics versus multi-omics with a visual. Imagine you are trying to understand how a really bustling, chaotic city works.
Okay, I like that.
A single omics approach, like just looking at the genes, the transcriptome, it's a lot like looking at a static map of the city's roads.
Right, it tells you what infrastructure is there, what paths exist.
Exactly. But it doesn't tell you if there's a massive traffic jam on Main Street. Multi-omics, which is what this study uses, is like having that map of the roads and simultaneously having access to the real-time live traffic data.
The proteins, basically, the proteome.
Yeah. By looking at both the roads and the traffic at the exact same time, you get a much richer, much more accurate picture of what is actually happening on the ground.
That map and traffic analogy captures the essence perfectly, because the genes give the structural instructions, but the proteins are the vehicles doing the actual work.
Makes total sense.
So to get the simultaneous view, the researchers set up a very specific experiment. They used 9-month-old male APPPS1 mice.
APPPS1. Why that specific mouse?
Well, this specific strain of mouse is a classic model in Alzheimer's research. It's been genetically engineered to have a very stable, predictable buildup of amyloid-β plaques.
Okay, so they know exactly what disease timeline they're looking at.
Exactly. At 9 months old, these mice are well into demonstrating the disease characteristics. And the researchers zoomed in on the hippocampus, which is the brain's primary memory center.
And one of the first regions devastated by Alzheimer's, right?
Yes, exactly. So it's the perfect place to look.
Okay, so if I'm a researcher with that disease tissue, how do I actually see the roadmap and the traffic? Like, how do these tools work in the actual lab?
Well, they use two highly advanced techniques. First, for the roadmap, they use transcriptomics, specifically RNA sequencing.
Okay, RNA sequencing.
Yeah. This essentially takes all the messenger RNA in the tissue, the temporary copies of DNA being sent out to build things, and reads them letter by letter. It lets them see which genes are being turned up or turned down.
So that's the roadmap. What about the traffic?
Right. For the traffic, they used a technique called DIA proteomics. That stands for data independent acquisition.
Data independent acquisition. Okay.
Yeah. And instead of just looking for a few specific proteins they already know about, this method basically shatters all the proteins in the sample into tiny fragments called peptides.
Oh, wow. all of them.
All of them. And it measures the mass of everything simultaneously. It's like taking a high-resolution panoramic photo of the entire cellular environment.
That's incredible. So they run the sequencing, taking these panoramic photos of both the RNA and the proteins. What was the initial haul when they compared the Alzheimer's model to the healthy brains?
The initial numbers were pretty substantial. On the RNA side, they found 263 differentially expressed genes.
Wait, 263. That's a lot of genes changing.
It is. Specifically, 162 were upregulated, meaning their activity was abnormally high, and 101 were downregulated.
Okay, and the proteins?
On the proteomics side, they found 448 differentially expressed proteins. Their 178 were upregulated and 270 were downregulated.
Okay, so we have hundreds of changes on the genetic roadmap and hundreds of changes in the protein traffic.
Yeah.
And here's where I really want to push back on the methodology a bit, because I read this part. The researchers cross-referenced these two massive lists, right?
They did.
And they found an overlap of exactly 6 targets.
Yes. 6 targets.
Just 6. Out of hundreds. I mean, 5 were co-upregulated and 1 was co-downregulated.
Right.
If I'm looking at 2 data sets supposedly describing the exact same disease in the exact same tissue, and only 6 things match out of hundreds, my first thought is, the data is broken.
Right.
Why is that overlap so incredibly tiny?
It's a completely natural reaction to look at that bottleneck and assume something went wrong. But the limited overlap isn't a flaw in the study. It's actually a reflection of how chaotic cellular biology really is.
Chaotic how?
Well, you have to account for post-transcriptional regulation.
What does that actually look like inside the cell?
Okay, so just because a gene sends out a piece of messenger RNA, it doesn't mean it automatically becomes a functioning protein.
Right.
The cell's environment can intercept that message. It can modify it or even destroy it before translation ever happens.
Oh, interesting.
Yeah, and furthermore, proteins and RNA have entirely different lifespans. Some proteins are incredibly unstable and degrade within minutes, while the RNA instruction that built them might stick around for hours. Or vice versa.
They're moving at different speeds.
Exactly. You're trying to take a snapshot of 2 moving targets that travel at totally different velocities.
Okay, so the translation from a gene to a protein is less like an architect handing a blueprint directly to a builder and more like a game of telephone in a really noisy factory.
That's a great way to put it.
The instruction gets sent, but a dozen things can happen to it before the actual machine is built.
Exactly. And that chaotic factory floor analogy explains why the researchers didn't panic when they only saw 6 perfect overlaps.
Dissected it.
Right. Instead of obsessing over individual one-to-one matches, they zoomed out and looked at the broader biological pathways.
And what did they find there?
They found that both datasets were clustering heavily in the exact same areas, specifically the immune system, metabolic pathways, and the complement and coagulation cascades.
So even if the specific parts don't match, the departments they belong to are exactly the same.
The individual steps might look different, yeah, but the overall biological signals were pointing in the exact same direction.
Okay, that makes total sense. Now, if you're listening to this and trying to keep track of the alphabet soup of gene names, don't worry. You don't need to memorize them all, but we do need to divide them into 2 distinct buckets.
Right. The knowns and the unknowns.
Exactly. The ones we expected and the ones that completely shocked the researchers. These are our magnificent 6, the overlapping targets that showed significant changes across both the RNA and protein levels.
And just to list them out, we have APP, LY86, C1QB, EGFL8, and CD180 as the co-upregulated one.
Right, the ones that went up.
Yeah. And ERMN as the single co-downregulated target, the one that went down.
Let's start with the familiar faces first.
Sure. The familiar faces basically validate that the experiment actually worked. APP, for instance, stands for amyloid precursor protein.
Which is like the classic Alzheimer's gene, right?
Arguably the most famous one, yeah. It's directly responsible for the formation of the A-beta plaques that choke the neurons. Seeing it upregulated is basically the biological smoking gun.
Okay, what about the others?
Then you have C1QB. This is a crucial gear in the complement system.
Okay, I know the complement system is part of our immune defense generally, but how does it function in the brain?
Think of the complement system as the brain's garbage disposal and tagging mechanism. In a healthy brain, it tags dead cells and debris so immune cells can come and clear them away.
Sounds hopeful.
It is, until it isn't. In Alzheimer's, the system essentially goes rogue. It starts inappropriately tagging healthy synapses, the actual connections between neurons, for destruction.
Oh, wow.
Yeah, and C1QB is a key initiator of that destructive process.
That's brutal. And the last familiar one.
Finally, for the known quantities, we have Ly86, which is an inflammatory regulator. Its expression is heavily tied to the worsening of plaque pathology.
Okay, so APP, C1QB, and Ly86, they prove the foundation of the study is solid. But here is where this deep dive gets deeply fascinating.
The novel genes.
Yes, half of this list, EGFL8, ERMN, and CD180, are completely novel in the context of Alzheimer's. Let's talk about EGFL8. Previously, this gene was known for playing a role in various cancers and in brain white matter issues, but it had never been linked to Alzheimer's until now.
It's a massive finding, primarily because of this weird nuance hidden in the data. EGFL8 was significantly upregulated in the Alzheimer's brain tissue. But when the researchers tracked its progression, there was absolutely no correlation between the expression of EGFL8 and the actual accumulation of the amyloid-β plaques.
Wait, hold on. The plaques are the defining destructive feature of the disease. How can a gene be highly elevated in a diseased brain but completely ignore the plaques?
This is exactly what suggests Alzheimer's isn't just a plaque problem, it's a multi-system failure.
Oh, I see.
EGFL8 appears to be driving pathology through completely parallel mechanisms. The researchers hypothesize it activates specific downstream signaling pathways, like the PI3K ACT or MAPK pathways.
Okay, alphabet soup again. Can you break those pathways down? What do they actually do?
Absolutely. The PI3K ACT pathway essentially acts as a cell's internal survival and growth switch.
A survival switch.
Yeah. It tells the cell how to use energy and whether to stay alive or undergo programmed cell death.
Okay, and the other one?
The MAPK pathway operates similarly, regulating how a cell responds to stress. If EGFL8 is inappropriately flipping those critical survival switches inside the neurons, it is causing profound cellular damage completely independently of whatever the amyloid plaques are doing outside the cell.
This is that saboteur from our hook at the beginning. It has nothing to do with the main assembly line breaking down, but it's pulling wires in the control room to shut the whole factory down anyway.
That's exactly it.
Crazy. What about the single downregulated target, ERMN?
So ERMN was found in significantly lower quantities in the diseased hippocampus, and what's compelling here is its medical history.
Right.
ERMN has previously been linked to major depressive disorder and autism spectrum disorder.
But on the surface, psychiatric and developmental conditions sound entirely unrelated to a physical neurodegenerative disease like Alzheimer's. How do they even connect?
It comes down to a process called genetic methylation.
Methylation.
Yeah, which is essentially the cell's way of putting a do-not-read sticky note on a gene to silence it. In depression, autism, and now Alzheimer's, we see similar defects in how genes like ERMN are being silenced. By improperly turning off ERMN, the brain is losing a critical layer of its baseline immune defense.
Wow.
Yeah, it shows a shared biological vulnerability across conditions we used to think were completely unrelated.
That is wild. And that leaves our final novel gene, CD180.
CD180 has a strong history of being associated with systemic inflammatory diseases.
So whole body inflammation.
Exactly. And finding it heavily upregulated here is a massive puzzle piece. It further proves that chronic neuroinflammation isn't just a symptom of Alzheimer's, but a core driver of the disease itself.
Okay, but finding a saboteur gene like CD180 in a computer database is an amazing first step. But a computer simulation isn't a living brain. How did they prove to the rest of the scientific community that these genes are actually misbehaving in real physical tissue?
Well, validation is an absolute necessity here because high-throughput omics data can sometimes produce false positives. To prove these digital signals were physically real, the team used two classic gold standard laboratory techniques. For the RNA, they used qPCR, which essentially takes a tiny invisible genetic signal and chemically amplifies it millions of times until it's easily measurable.
Okay, and for the proteins?
For the proteins, they use Western blots. That involves using specialized antibodies that act like homing beacons. They physically latch onto the specific target proteins so researchers can actually see them.
They also didn't just look at one isolated moment in time, right?
Yeah.
They utilized a database called Mosaic. Why does chronological tracking matter so much here?
The Mosaic database is a phenomenal resource. It allowed the researchers to track gene expression profiles in mice from 2 months of age all the way to 18 months of age.
So you get the whole lifespan.
Exactly. You can literally watch the biological timeline unfold, and it showed strong positive correlations. As the mice aged and the amyloid-β plaques heavily accumulated in the terminal stages of the disease, the mRNA levels of genes like C1QB, CD180, and Ly86 spiked right alongside them.
So they move together.
Yeah, it firmly anchors the genetic markers to the physical progression of the disease.
Now, I read the methodology closely, and there's a statistical nuance I have to bring up here.
Oh, sure.
When they did the Western blots for LY86 and ERMN, the results didn't hit formal statistical significance, even though the advanced DIA proteomics sequencing said they were highly significant.
Right, I know what you're referring to.
If the physical test fails the math, doesn't that throw a wrench in the findings?
It's a very sharp observation, but it doesn't invalidate the findings. It really just highlights the difference between an analog tool and a digital one.
Oh, so?
While a Western blot relies on a single antibody binding to a protein, it can be pretty finicky. It's subject to subtle variations in how efficiently that one specific antibody attaches.
Okay.
DIA proteomics, on the other hand, quantifies multiple different peptide fragments of the protein simultaneously. It is vastly more sensitive at detecting moderate changes.
Ah, so it's just a better magnifying glass.
Exactly. The crucial takeaway is that the biological trend, the actual physical direction the protein levels moved, was perfectly identical across both methods. The compass needle pointed north on both instruments. The omics sequencer was just precise enough to prove it mathematically.
Got it. Okay, let's talk about application. We have these verified biological targets. How does understanding these specific molecular switches translate into helping the millions of human beings actually affected by Alzheimer's?
The researchers took a very clever approach to bridge the gap between the lab bench and the pharmacy. They took their 6 identified targets and cross-referenced them against a database containing 27 FDA-approved Alzheimer's drug targets.
Oh, that's smart. What did they find?
The results were stunning. 4 of the 6 identified genes, CD180, LY86, C1QB, and APP, encoded proteins that directly interact with those known approved drug targets.
Wait, 4 out of 6? That is a 66.7% hit rate.
It's an exceptionally high strike rate. It tells us that these novel biomarkers aren't just floating around in biological isolation. They are deeply entangled in the specific molecular networks that our current best drugs are already trying to manipulate.
That's amazing.
Take LY86 and CD180, for example. The data shows they are key regulators in something called the TLR4 pathway, which controls inflammatory responses.
So it's like we've been trying to put out a fire by solely spraying water on the smoke, the plaques. But targeting the TLR4 pathway is like finding the broken gas valve that's actually feeding the flames of neuroinflammation.
That is exactly the shift in perspective this research offers. Instead of just attacking the plaques directly, pharmacologists might be able to develop a brand new anti-inflammatory strategy.
Turning off the gas.
Exactly. By inhibiting the TLR4 pathway, you could shut off the gas valve, stopping the excessive immune response that physically degrades the brain tissue.
It's incredibly promising.
Yeah.
But we have to look at the blind spots. The researchers were very transparent about the limitations of their study, and one massive limitation stands out to me. They exclusively used male mice.
Yeah.
Considering that females make up the vast majority of human Alzheimer's patients, and that estrogen fluctuations are a known major risk factor for the disease.
That's a huge issue.
Relying entirely on male models feels like a glaring omission.
It is a profound limitation, and they acknowledge that. Sex-specific differences in neuroinflammation are really well documented in biology.
Right.
Female mice naturally show higher baseline levels of pro-inflammatory cytokines, and estrogen plays a massive role in regulating immune responses in the brain. By only using males, which they likely did just to keep the baseline data stable for an early exploratory study, they have undeniably introduced a bias.
They could be missing half the picture.
Exactly. They might be completely missing inflammatory markers or mechanisms that are uniquely critical to female patients.
And the APPPS1 mouse model itself has inherent limitations, doesn't it? Like, it has the plaques, but it doesn't really replicate the full human experience of the disease.
Far from it. The mouse model aggressively overproduces amyloid-β plaques, which is great for studying that one specific feature. But human Alzheimer's, specifically sporadic late-onset Alzheimer's, is infinitely more complex.
What's it missing?
Well, this mouse model completely lacks tau pathology. Those are the destructive tangles that form inside human neurons.
Oh, that's a big deal.
It is. And even more importantly, it lacks the significant widespread brain cell death we see in human patients. I mean, a mouse lives in a controlled box for a few months. It doesn't experience the complex interplay of 80 years of aging, environmental toxins, and varied genetic risks.
Which leaves us with a final provocative thought to mull over today. If our premier animal models for Alzheimer's fundamentally lack the complexity of human aging, completely miss crucial pathologies like tau tangles, and are heavily skewed by sex, Are we struggling to cure the disease simply because we've been testing our treatments against an incomplete reflection of it? It's a question that demands our attention as science pushes forward into the multi-omics era. This paper is published under a Creative Commons Attribution 4.0 license, and full credit is provided in the episode description. Thank you for listening to One Paper Orbit, our research paper podcast. Our goal is to make peer-reviewed research accessible to everyone. If this episode was helpful, please share your thoughts and let us know which papers you'd like us to explore next. Thank you.